So this is in line with the last abstract. So this is a CAR T-cell product that was initially sort of developed at the Medical College of Wisconsin, and it used a tandem anti-CD20, anti-CD19 CAR T-cell targeting modality. And the idea was similar in the sense that we wanted to target more than one B-cell antigen to see if that can improve efficacy. But additionally, as part of a phase 1 clinical trial we did at the Medical College that sort of led to this study, we had also learned that maybe giving a fresh infusion rather than the traditional freezing the product down and then thawing and administering it may actually lead to improved outcomes, but it also allows for rapid CAR T-cell delivery. So based on the data that was previously published, we worked with a sponsor to develop a multi-center phase 2 clinical trial to use the tandem anti-CD20, anti-CD19 CAR T-cell product in third-line diffuse large B-cell lymphoma. So these are relapsed refractory patients that are generally chemotherapy refractory, and we use this model of an automated manufacturing system where the cells would come in fresh, be delivered fresh actually to the treating institution. And this meant that lymphodepletion started during the manufacturing process to facilitate that fresh infusion. The advantage of this approach is that patients got the CAR T product truly in a 14 to 14 day vein to vein time, because again, the target was a fresh infusion. So this was an interim analysis of this CAR T-cell product with this novel manufacturing platform looking at a pre-planned sort of efficacy endpoint. So we looked at 59 evaluable patients, and we found that the overall response rate was 73% in a patient population that had high IPI, high LDH, and was otherwise a poor group of patients with the relapsed refractory DLBCL. The CR rate was about 51%, and the 6-month progression-free survival was 55%. Like other trials, this is an ongoing study, so there's patients still being accrued, hence the follow-up is limited, and we look forward to reporting 1 and 2 year efficacies as that data becomes more mature. In terms of safety, a big question is, if we target both antigens, are we going to see a differential toxicity profile? And the good news is that did not play out. In fact, the toxicity profile was impeccable. There was no grade 3 or higher cytokine release syndrome. That's a type of cytokine release syndrome where patients can get quite ill. All of it was grade 1 to 2, which was very manageable. Similarly, grade 3 or higher ICANS was also low at 4%. And again, very manageable, with the majority having grade 1 to 2 toxicities. So as part of this pre-planned interim analysis, I think we showed a few major points. 1, it is feasible to do a fresh in and fresh out CAR T. 2, that a tandem CD20, CD19 CAR T is efficacious and safe. And 3, we can deliver this in a rapid timeline. And that really improves the accessibility part to patients. No patient died waiting for this CAR T-cell product. But again, they're starting chemotherapy shortly after apheresis and then getting a fresh product delivered in a timely fashion. So we're very excited about this. We think that this could be potentially a paradigm shifting model on how to do CAR T. We're looking forward to the completion of this study and reporting the final results at a future Congress.